Listen-only mode.
Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Treatment Allocation in Randomized Trials, Challenges and Best Practices. My name is Nima, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank IDDI, who helped to develop the content for this presentation. The International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Raleigh, and San Francisco. Now I'd like to introduce our speakers for today's event. Dr. Everardo Saad has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology, and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. Ms. Linda Danielson has been at IDDI since 2006, overseeing all the national teams at IDDI, including randomization, data management, statistics, and project management. She co-authored a chapter on randomization in adaptive designs in the book Practical Considerations for Adaptive Trial Designs and Implementation, edited by Wele Hay, Jose Pinheiro, Olga M. Kuznetsova in Springer 2014. Ms. Danielson is interested in clinical trial design, statistical methods for clinical trials, randomization methods, and increasing efficiency in clinical trials through web-based systems. She holds a master's in biostatistics from the University of Wisconsin-Madison. And now, without further ado, I'd like to hand control over to our very first speaker. Dr. Saad, you may begin when you are ready.
Thank you, Nima. Hello to all of you. Thank you for attending this webinar. So Linda and I will talk about randomization, and I will start with the more scientific or conceptual issues about the methodology and some of the methods that we use for randomization, whereas Linda will cover more practical issues regarding the implementation, including system requirements, drug supply requirements. She will show us some case studies and end by showing us suggested best practices. Now, let me start very simply by reminding all of us why we need randomization. The three first bullets are more obvious when we think about such need. For example, randomization provides a sound basis for the use of statistical methods. They remove selection bias by allowing comparisons of like to like, so to speak. We often think about known prognostic factors in such comparisons, but perhaps more importantly, unknown prognostic factors are hopefully balanced when we use randomization. And also, it's important to distinguish association from causation, and the only way to do it with reliability is by using randomization. Now, there are other reasons why we may want to randomize, and this is very important for certain types of clinical trials and scientific projects. For example, if one wants to untangle the prognostic from the predictive role of biomarkers, it's important to randomize. Otherwise, you can't tell whether a biomarker is just prognostic or also predictive in the sense of allowing for choosing a certain treatment for patients. To validate surrogate endpoints, you have to have randomized evidence. Otherwise, you cannot go about doing it, as we have shown in a recent webinar in this series. And also, in order to avoid conclusions based on poor surrogates, it's important to randomize. And I'm going to show you an example of what I've just said in the last bullet of the previous slide. So until 2003 in oncology, there was very strong evidence that correcting anemia in head and neck cancer patients by using this growth factor, erythropoietin, would improve the outcomes of radiotherapy, because indeed it was known that this outcome related to oxygenation of the tumor at the time of radiotherapy, and by increasing the hemoglobin levels, oxygenation would increase, and then treatment outcomes would be improved. And that was the prevailing thinking at the time. And the drug was used relatively frequently until they really decided to run a randomized trial, which had as the primary endpoint overall survival. So remembering that until then, hemoglobin levels had been used as a proxy for the effect of the drug. And interestingly enough, the study showed a statistically significant difference in overall survival, but favoring placebo and not the drug. So this shows why randomized evidence is important. Otherwise, we may get to wrong conclusions, especially based on a poor surrogate outcome. Now, more recently, all of us are really very enthusiastic about precision medicine, which is the attempt to give very targeted therapies to patients with the correct biomarker. And we often see situations such as this one shown on this slide, where you have patients that were treated with targeted therapy aimed at modulating some very specific abnormality that they have in their tumors, and then they are compared with patients without such abnormalities in light blue, or patients with the abnormality but not using a targeted therapy. And we're very often impressed by, for example, this difference in survival probability, which is statistically significant. But this comes from a retrospective analysis. It's not a randomized study. And in fact, the randomized study that was done in order to try and validate this concept, it's one of the studies that are planned for this type of situation, comparing targeted therapy, which is the blue curve here, with chemotherapy, which is the red curve, showed no difference whatsoever in the primary endpoint, which was in this case progression-free survival. So the concept that appears nice, as shown in the previous slide, when tested in a randomized trial may not prove to be correct, or at least more evidence is required before we accept some interventions without randomized evidence. And for this reason and others, our group very recently wrote this paper in which we try to argue that even with precision medicine, which is very compelling in terms of having the right drug for the right patient, but even in that situation, randomization makes sense. And in this paper, we try to argue that randomization is needed in the various phases of drug development, beginning in phase 1. For example, in this day and age of expansion cohorts, it's an interesting idea to start randomizing as soon as we have a dose level that is considered safe, and try to expand that cohort to have a look at the activity of the drug. So more and more this is done in phase 1 studies already, as we know, and we propose that this should be, whenever feasible, done under randomized contrasts, preferably having a standard of care as the control arm. If a standard of care is not available, any other treatment than the investigational therapy may be used, and we would very much favor this approach as long as early stopping rules are in place, for example, for outstanding activity of the new drug, and preferably with analyses that are based on biomarkers and that can guide decisions during the drug development process. This more or less goes for phase 2 as well. In phase 2, it's more historically more common to randomize, and randomized studies are increasingly more frequent, especially in oncology, but more and more we believe that they should be randomized and trying to get as much as possible evidence on the activity of the drug, especially when there are discussions about whether it will be ethical to conduct a phase 3 study if a drug is very active. So one of the reasons to randomize early on is to make sure that whatever promising activity is observed is really due to the drug and not to the prognostic role of the biomarker. And in phase 3, of course, randomization is almost inherent in the definition, but more and more it's important to include a true control, which can be not very often in oncology, placebo or best supportive care, but this is feasible in many other fields. But in oncology or other fields where this is not feasible, for example, whatever available standard is in the market, or even treatment of physician's choice can be used as a control arm in phase 3 trials. And then early stopping for outstanding efficacy again can be implemented based on interim analysis, which can be conducted, for example, on surrogate endpoints. And this would be, of course, a topic for a different discussion. Now, let me briefly present some of the methods. These are not all the methods that exist for randomization, but they are certainly the most common ones and the ones that are usually discussed, and usually the decisions are around choosing one of these methods as they will be presented, or with some slight modification. So let me start by showing you what's called simple or unrestricted randomization, which is conceptually the most robust method, because it's really a play of chance, and it's analogous to flipping a coin. Of course, in practice, this is best done using a computer program, but it's analogous to flipping a coin between heads and tails if it's, for example, a one-to-one randomization. So in statistical terms, it's quite a robust method, but it can be problematic in small studies, because one can have imbalances with regard to certain prognostic characteristics. So unless a trial is very large, we do not advise, and neither do other people in this field, to use simple randomization. Now, the question, of course, is what is large? No one knows exactly. Probably more than 1,000 patients, and only in some disease areas do we have the luxury of having such large studies. Now, this is just to illustrate the imbalance that would be present depending on the number of prognostic features for which you want to achieve balance, and considering the probability of severe imbalance. And we defined severe imbalance here as any dichotomous characteristic that would be distributed in a ratio that is more than 60 to 40. So ideally, it should be 50 to 50 between both study arms. And we consider severe if arbitrarily if it's more than 60 to 40 distribution. So according to the number of characteristics and according to the trial size, which is indicated by the different colors, you have very large imbalances in terms of at least one of these characteristics. And so this is just the empirical sort of demonstration why simple randomization is not very often done, especially when many prognostic factors have to be taken into account. Now, so in order to compensate that, the most common alternative is to use some form of stratification, which is often advised. So we consider the most relevant prognostic factors and then randomize patients within strata according to the cross classification of such prognostic factors, typically considered as dichotomous. So for example, in this study here, the randomization was stratified according to the type of non-Hodgkin lymphoma, follicular versus others, the type of refractoriness that the patients had before the trial, so they could be refractory to either monotherapy with rituximab or to rituximab combined with chemotherapy, another dichotomous factor. Then the number of previous therapies, less than or equal to 2 versus more than 2, and then according to the geographical region. So these are 4 stratification factors. The cross classification of these factors leads to different strata, and patients are randomized within these strata. Now, one of the problems with this, with pure stratification, so to speak, is that we can only use a relatively small number of factors. Otherwise, there will be too many strata, and this will create problems for statistical analysis. So another important feature often used is permuted blocks, which helps to ensure similar numbers of patients across trial arms as the trial moves along. So this is especially important early on in a trial, because at any given point in time, we try to ensure that a similar number of patients will have been randomized to the 2 groups being compared. This is easily implemented within the strata, so these 2 things can go together, permuted blocks and stratification. But here sometimes it's argued that depending on the block size, it can lead to investigators to sort of predict or guess what the next assignment will be, and this may create arguably some kind of selection bias by selectively including or not including patients with certain features if the investigator knows what the next assignment will be. Now, we at IDDI very much favor minimization as the preferred method whenever possible. One of the problems with stratification is that, at least in some disease areas, for example, in oncology, the center is very often an important prognostic factor. And in a multi-center study with many centers, it may be difficult to stratify for each and every center. So one of the solutions is to use minimization, because it allows for using as many factors as needed. And then I will explain in the next slide how it's done. And the idea is to minimize the imbalance in prognostic features along the study between the 2 study arms. And this is done by checking at after each patient is screened, what would happen if this patient was included in one arm versus the other, and trying to minimize the imbalance that would result from the inclusion in arm A or B, say, and this is done by using this method. So it's one of the so-called dynamic allocation methods, because there is no randomization list that is followed, so there needs to be an algorithm that is constantly checking for each and every patient the prognostic features and then allocating the patient to a certain group. Now, this is sometimes criticized because people might think that the allocation is deterministic, but in fact, minimization is very often implemented using a stochastic component through which every patient that enters the study is in fact randomized, but not in a 1-to-1 ratio. So the probabilities are not 50 to 50%, but rather some kind of imbalance favoring the arm where there will be a better balance in prognostic factors. At IDDI, we often use 80 to 20, but other ratios can be used depending on the situation. So let me give you one example. The top part of the slide shows the distribution of patients per arm in a study, a fictitious study, let's say comparing drugs A and B. And then let's say we identified 3 prognostic factors, age, gender, and institution. So let's say age has 2 categories, and the categories that we're looking at is between 50 and 65 years. 55 and 65 years. The gender we're looking at is male, and the institution, let's call it XYZ. So let's say at this point in the study, 94 patients have been randomized to arm A and 96 to arm B. And this is the number of patients with each of these prognostic features in each of the study arms. And now another patient comes in and is eligible to the study. And let's say it's a 60-year-old man in site XYZ. So what the algorithm does is to first pretend the patient will be randomized to arm A. So 23 patients with that age had been randomized. If we randomize this patient here, it will be 24. And then the imbalance will be 24 minus 22. It will be 2 patients. Now, again, the algorithm pretends the patient will go to arm B. And then if the patient goes to arm B, it will be 23 patients with this age in arm B. And so the imbalance will be 23 minus 23. It's going to be 0. So the algorithm does it for every single prognostic factor. And then it sums the overall imbalance that would be achieved had the patient been randomized to A or B. And then it takes the arm for which there would be less imbalance, and this would be arm B, because there would be an overall sum of 5 versus 7. And so this patient will be randomized with a higher chance to arm B than to arm A, but there's still a chance that the patient will go to arm A, but we use, like I said, a ratio that will favor arm B in this case, and so eventually in the long run things will balance out, and we can do that for many prognostic factors, even in a small trial. Now, like I said, this is one of the types of dynamic allocation, and there is another type which is very much discussed nowadays, called outcome adaptive randomization. So the previous randomization type, minimization, it's dynamic, but it's based on prognostic factors. In other words, on baseline factors. This one that I'm showing now is based on things that happen after the patient entered the study. So depending on the response, for example, that the patient has to the treatment, or that the patients have had at a certain point in time, the algorithm will change the ratio of allocation based on such response. So more patients will go to arm A or B, depending on the previous response of patients that have entered the trial already. This might sound like a very intuitively good thing to do, because you're in a way favoring patients entering the arm with better results, but there are many statistical problems. This is very well described in many papers nowadays. Some people argue it's more ethical, but many other people argue it's not more ethical, and there is a whole issue of this journal called Clinical Trials with several papers written discussing the pros and cons, and we at IDDI very much are against using this method of randomization. If one wants to have more patients in an experimental arm, for example, to have more information about this new treatment, we very much favor what's called unbalanced randomization, which means instead of, if minimization is not used, let's say just stratification with permuted blocks is used, instead of using a one-to-one randomization, one can do different ratios. The more often used is a 2-to-1 ratio, favoring the experimental arm. So 2 patients go to the experimental arm, to every patient that goes to the control arm. And by doing that, especially in early phase studies, smaller companies, for example, can get more information about their drug. At the same time as they don't need to have so much information about some kind of standard treatment that will be given to patients in the control arm. And so we think this is very useful, and we often advise our clients to do that. This causes a very minor loss of power. For example, for a 2-to-1 randomization, it's in the order of 10 to 15%, larger sample size. So this small increase in sample size, we believe, is more than compensated by getting more information on the new drug. And this is just one example where this was done in a drug that's approved for the treatment of lung cancer. And this was a phase 3 trial, by the way. This is often done in phase 3 as well, and we very much favor it. It would be, according to some people, a much better alternative, for example, than outcome adaptive randomization. Now, some of you may know the consort statement, which is a set of criteria and a checklist for publishing or rather for reporting the results of randomized studies. Consort stands for Consolidated Standards of Reporting Trials. And in this checklist, there are many aspects of the study that need to be described. And about randomization, at least 3 aspects need to be described. This is part of the transparency that is more and more required of investigators when they report the studies. So investigators need to make it clear what method they used to generate the sequences of randomization, the type of randomization, for example, whether any restriction was applied, such as permuted blocks or other types of restriction, such as stratification, for example, and then how they made sure the allocation was concealed throughout the study, what mechanism was used to make sure this would happen. And then finally, even implementation is considered. So who generated the random allocation list, who enrolled the participants, who assigned participants to these interventions. And in her part of the talk, Linda will cover some of these issues. So before I hand it over to Linda, let me just quote from this famous book about experimental designs, where the authors say that randomization is analogous to insurance in that it's a precaution against accidents, in scientific terms, biases that may occur and that are serious if they occur. And we would add that it's true, insurance protects against unlikely accidents, but randomization is even more important because it protects against biases that are almost certain to occur when it's not used. So I will now pass the podium to Linda.
Thank you very much, Eduardo. So I hope that you are now all convinced that you need to randomize your trials, and you also have learned about some different methods of randomization. So I'm going to talk about some things you need to think about when you're implementing a randomization system. First of all, as in all aspects of clinical trials, there are several different guidance documents. There are 2 guidance documents from the FDA, which are a bit older now, but they're still being followed not only for IRT systems, but for any type of computerized systems in clinical trials. There's one in May 2007 from the FDA, and the very well-known 21 CFR Part 11 from 2003. Another guidance that's a little less known is one from the European Medicines Agency that came out in December 2013, which is a reflection paper on the use of interactive response technologies, or IRT. And I'm going to talk about some of the things that are in that document. So that document mentions a few requirements for implementation of an IRT system. The first thing, because we are talking about a randomization system which will randomize the subjects, as well as providing study drug to the patients in the study, we have to define the access permissions. There are different types of users of the IRT. There's the investigator, who's going to be randomizing the study, doing the randomization, and also seeing the patients, allocating. The pharmacist, who might be an unblinded person using the system, the monitor, and the sponsor. So we have to define who's blinded versus unblinded. Also, if it's a double-blind study, the investigator must be able to unblind the patient in an emergency. And usually this should be someone with a medical background in order to do this unblinding. And good practice says that this investigator must be able to do this if there's an emergency without having to contact the sponsor, and ideally directly through the system. As such, the system should be accessible 24 hours a day because we have clinical trials all throughout the world, and it has to be able to cover every single time zone. And no system is absolutely perfect. It should be as perfect as possible, but we do need to have a disaster recovery system and backup systems in case there are any issues. And if at the site, for example, the site has no internet access for some reason, they have to be able to contact the IRT provider in order to do a manual intervention to randomize a subject, or even more important, to allocate drug to a patient if they come in to their visit. If this manual intervention is done, it must be documented and the system must be updated afterwards so we know exactly who got what treatment, and in case of a manual intervention, who did it. The system itself needs to have a very accessible audit trail which shows who does what, who randomized the subject, when did they randomize the subject, if there are any changes to the data, why did it change, and who made the changes. And all data in the system must be updated in real time. In addition to system requirements, there are drug supply requirements. The IRT system must ensure that sites have the appropriate medication and also that subjects do not run out of medication. In order to do that, the IRT system needs to think about, well, it has to be set up so that it thinks about time for shipments to reach different countries, because we also know that some countries have very long importation times and it could take a while before the drug actually reaches the site. When allocating the drug, dates have to be taken into account, the date of the expiration of the drug. So, for example, if the patient goes home with their packet of medication that should cover 2 weeks, the medication can't expire within that 2-week period. It has to be good for at least a week over, depending on how long they come back to the site. So that is what we call the do not dispense date. In addition, in order to ship the drug from the depot, we need time for it to get from the depot to the site. It's at the site, it's given to the patient. So we have a date that's called the do not ship date, which is even more before the expiration date in order to ensure that no patients receive drug that could expire when they're using it. So these are requirements that we need, but we also know that we might sometimes have to make changes during a study. We might have to modify the protocol. We might have to make some changes, especially in early change studies. And if we are going to make changes during the study, we need to do very careful thought and assessment to these changes and what impact they might have onto the IRT system and to the randomization itself. Anything such as adding stratification factors or treatment arms have to be considered very carefully. And the ability to amend the central allocation system should be assessed at the time of the vendor selection. The sponsor needs to think about the randomization technique. Will this allow this change to be made? And does the IRT provider have the ability to amend the system? Because this does happen, as I said, especially for early stage studies, I have 2 case studies which show how we at IDDI have been able to do this. So the first case study is a 4-arm phase 1 study in ophthalmology. The study had very slow recruitment, and the sponsor was concerned that they were not going to be able to recruit all of the patients in the study. So a decision was made to widen the inclusion criteria, to include additional anti-VEGF treatment and also a different treatment schedule. Both the previously used anti-VEGF and the treatment schedule could be considered as prognostic factors. They are two factors that could influence the response of the patient. So that's why those factors would be included in the statistical analysis, and the sponsor also wanted to include this in the randomization. So we had to have a look at it and to determine if it would be possible. Fortunately for this study, all of the subjects up to the time of that protocol amendment were using one specific anti-VEGF treatment, which I'll call E, and they had one treatment schedule, which I call 1. So at the time of the amendment, we could put all patients who were already randomized into one specific strata, and then we could update the minimization so that after that we could ensure the balance in each stratification factor. Just to kind of show what this looks like, before the amendment, you can see we have Schedule 1 and anti-VEGF E. There were a total of 16 patients randomized, and there were 4 patients in each treatment group. So that was great, and all of the other factors were empty because until the time of the amendment, that was not allowed by protocol. 1 year after the amendment, we still had the majority of patients in this group. As you can see, it was very well balanced with between 14 and 16 patients randomized to each treatment group. The Schedule 1 with the L also had a very good balance, as did Schedule 1 with A. Now, I do want to make a comment here because you can see that Schedule 2 with L and Schedule 2 with A have almost no patients. But what's important to know is the minimization ensures that we have a balance within each stratification factor across the treatment groups. But it was not a capped value where we tried to force patients into one group or another, because in any case, they came into the site and they were already using either Schedule 1 or Schedule 2, and they were either using anti-VEGF E, L, or A. So at the very end of the study, there still were the majority of patients who were in stratification 1E, but as you can see, that stratification factor was well balanced across the study, across the treatment groups, as were the other stratification factors. And in total, we had a very nice balance. And in case we wanted to see, for example, was Schedule 1 well balanced and was Schedule 2 well balanced, we can see that we were able to keep that balance as well. We can also see that adding Schedule 2 did not really add a large number of patients to the study, but it did bring a few more extra in so that the study could stop randomization on time. If we look at the total within each anti-VEGF group, we can also see that that is very well balanced. So that is a big advantage of using minimization, is that it takes the overall balance into account and also within each of the groups. So for case study 2, it was a study in oncology, and it was a phase 2 study, an early phase 2 study. So as Alvaro mentioned, sometimes it's good to have studies like that have an unbalanced randomization. So we had a 1-to-2 randomization. Standard was 1 and 2 was treatment. So we were putting more patients onto the new treatment. There had been 30 patients randomized, 11 in arm A and 19 in arm B. Well, a decision was made to add arm C, which is actually a combination treatment of arm A and B. And because they wanted to see, and they weren't completely convinced that arm B was the best way to go, they wanted at the end of the study to have a 1-to-1-to-1 balance among the 3 treatment arms. If we had just added arm C, minimization would have taken care of making sure that we had a balance. But in fact, what would have happened is almost all patients would be allocated to C until C caught up. As I've already mentioned, we don't use deterministic, so it wouldn't be sure that they would all be allocated to C. But if we didn't do anything different, 80% of the patients would be put into arm C at the time of the amendment. That was not what we wanted to do. And so we looked at the total number of patients to enroll, which is 126. So actually at the end of the study, we wanted to have 42 patients in each treatment arm. So what we did was we actually set a very unusual looking balance of 31, 23, 42. Well, that means that we had 31 because we had the 11 already included and we needed 31 more in that group. We had 19 already included in arm B and we needed another 23, and we needed 42 in arm C. In order for this to work, at the time of the amendment, those 30 randomized patients were no longer counted in the calculation. So we started at 0 again in order to get that balance. So what does that look like? At the time of the amendment, we had, as I said, 11 in A and 19 in B. After 6 months, we had 20 in A, so we had an extra 9 subjects were randomized to arm A, an extra 7 subjects to arm B, and 12 subjects to arm C. So after 6 months, we didn't have the balance, but we knew that we had to continue including patients in the study. After 1 year, we had 24 patients in A, 29 in B, and 17 in C. So it was almost caught up, but we still had quite a few patients to include in the study. And then just before the end of the study, we had 41 in treatment A, 42 in B, and 42 in C. So we were able to catch up and to have a nice balance at the end of the study. I do want to mention that this is a change that can be made in a phase 2 study because we're still exploring the different treatments. We're still trying to figure out what's happening with the drug. I would not recommend such a change to be made in a phase 3 study because it's a lot of changes while the study is going on. And talking about changes and things we need to think about, I'd like to end with some best practices for different systems. First of all, sponsors really have a lot of responsibilities. And these responsibilities are also mentioned in the reference guideline that I showed at the beginning of my section. First of all, the sponsor should really do an audit of the IRT provider. The sponsor needs to ensure that the provider can do the randomization and has everything in place that they need to do. They also need to ensure that the user requirement specifications match the protocol and also any special needs of the IMP. I mentioned the user profiles already. It's really up to the sponsor to work with the IRT provider to define the end user profiles and who can access to what in terms of blinding and unblinding. If there are any special activities to be done by the pharmacy, the sponsor needs to take care of that. And very importantly, the sponsor should perform the UAT. The UAT is user acceptance testing. So the sponsor should really use their own tests so that they can test independently of the IRT provider, and they need to include sponsor, investigator, and monitor roles. The last bullet was not mentioned in the guideline, but something that we at IDDI recommend is the sponsor should also consider whether their IRT system can be integrated with other study systems, such as the EDC or the drug supply distributor. If it can be integrated, it's much easier for the sites. They can log into one system and the other system, and that system can call another system. Part of what we need to have are specifications and validation. And the very first specification is actually in the protocol. We need to have a statement that randomization, is used, but what is the randomization scheme? How are we doing the randomization? And ideally in the protocol, we should also state that an IRT system will be used. The validation of the system must be in line with regulations. The URS produced by the vendor and approved by the sponsor. The system validated by the provider, of course. And any incidents found during that validation should be fixed prior to release and documented. At the provider, we need to have a review and sign off by an independent QA department. And in addition to that sign off, there should be a formal sign off by the sponsor prior to the first patient being randomized. As already mentioned, any protocol changes might result in system changes. And so there needs to be a change control procedure at the provider in order to ensure that any changes are made correctly. Now, I've talked a lot about the system, but in order to do the randomization, we need different types of lists. If it's not using minimization, but it's using permuted block randomization, we need a randomization list. And the aim of that list is balance within a block. But we also need a randomized kit list if it's a double-blind study. And that is needed in order to help make the kits unpredictable, keeping the blind. And for the kit list, the block size is not so important, but it can be useful to make a kit list with a certain block size. So at IDDI, we really recommend that the program for creating the randomized list or the randomized kit list is written by a statistician, because the statistician is the best one to ensure that it's statistically correct and it completely adheres to the protocol. But of course, the study team must remain blinded. The statistician is part of the study team. And in order to allow that statistician to remain blinded but have the correct list, we have a member of our randomization team run the program, and they use what we call a different seed. Now, the seed is something that's used by random number generators, and each seed value corresponds to a specific sequence of values. So if you provide the same seed twice, you get exactly the same sequence of numbers. So when our randomization team runs the program and they change the seed, by definition, it's a different sequence of numbers. So the list is, again, randomized, but the structure is exactly as it should be. So I mentioned the kit list, and the kit list has the kit number, which I say meaningless, but of course it's used. That kit number is what's used by the site. They know what kit number they're supposed to take. They take the kit number, the patient has the kit number, we record the kit number, but there's no order in that number. Whereas there's a sequence kit number that the site doesn't actually see, but the IWRS system and the drug distributor uses the sequence kit number. It's used to sort the list, it's used to store the kits, to order the kits, and to allocate the kits. There can also be a confirmation number or security code. Just to let you see what I'm talking about, you see here I have a kit list. So you see the kit number here at the left, and these are numbers, 5921, 6481, 4831. You can see there's no order in these numbers. You might be able to guess that this is a list created with a block size of 4, because after 4, we have a balance between active and placebo. And the sequence number is what lets us know which kits we need to take when. And then at the end, we have the end of the list. And so we still have very random looking kit numbers. And this helps so that the site doesn't think, oh, I've just taken kit 5921, so I need to take kit 5922 next. It's not at all like that. It's just a very random number. And as part of the checks that we put into place, we want to make sure that the kit list is being used correctly, that the randomization is working correctly. So we have statistical programming to validate the randomization, the algorithm itself, and also the allocation. And we have monthly checks of ongoing studies to ensure that the groups are well balanced and study drugs are properly allocated. Because in fact, none of us are perfect, and it happens sometimes a site might give the wrong kit. So for example, the system allocated kit 77302. So the study coordinator went to the refrigerator and took the kit, and they found the kit and they gave this kit. But if you look at the kit, you can see that it's actually kit 73302, which looks very similar, but it's not exactly the same. And so we recommend that there should be a distinction made between the kit allocated by the IWRS system and the treatment actually dispensed at the site. So if we have a place in the EDC where we can enter this information, we could see that the IRT allocated kit 77302, but we would know that the site dispensed kit 73302. So at the end of the study, we would be able to do a reconciliation of what was allocated versus what was dispensed. Now, it might be that that might be exactly the right treatment. We don't know yet exactly what's contained in the treatment, but that we know at the end of the study when it's reconciled. So finally, with the increasing complexity of randomized trials, we know that treatment allocation and randomization is more critical than ever. It requires a combination of reliability and agility, Based on advanced methodology and technology, there are different types of randomization we can use, and we need to have the system in place that can take care of these different systems. We need to make sure that the randomization scheme is tailored to the protocol. Ideally, we should integrate randomization with other systems to ensure smooth study flow. The IRT provider needs to have the ability to make changes during the trial, but also needs to be very careful about what types of changes can be made and how to put those changes into place. The validation of the randomization schema is very important, as well as validating the system. And there should be robust quality control of the randomization records during the study, so that after the study we know exactly who got what and when they got it and which kits they got after they were randomized. Thank you very much.
All right, well, thank you very much for that insightful presentation. We are going to be moving on to the Q&A portion of this webinar. So I have already received some questions from the chat panel from the audience, so let's begin with those. Would you recommend minimization in all multi-center studies? That is our first question.
Yes, definitely. We very much favor minimization over other methods, exactly because you can account for several prognostic factors, especially in oncology, where center is a prognostic factor. So I would say the more centers, the more we would favor using minimization.
All right, thank you so much for that answer. Our next question from the audience is it possible to implement changes in the randomization system without stopping active enrollment of patients?
Yes, that's a very good question. In fact, you can implement the system. What you have to do is all of that validation, the changes that I mentioned, have to take place on what we call a validation system. So first we create the changes, we validate the changes, we test the changes, and once all of those are correct, then we can put it live. So it can be, you can have the randomization up to 1 day before you put the new things into place, but it takes a lot of good communication between the sponsor and the provider to ensure that we can put this into place in an efficient way.
All right, thank you for that answer. Our next question is an unblinded trial, in an unblinded trial, is a kit list still needed?
I'll take that one as well. In fact, you don't necessarily have to have a kit list, but it can help in terms of the drug supply, because you can order by kit list, you can order by kit number. And also, we actually recommend, even for open label studies, to treat it as blind as possible, meaning that in the CRF, you don't want them to note that the patient's getting treatment A or B. It's better if they can, mention the kit number. And so if it's feasible and not too expensive, then I would recommend using kit numbers.
Okay, thank you for that answer. Our next question from the audience is minimization. Is it also recommended if the number of patients at each site are low, around 5 patients?
Yes, thank you for the question. I guess the person is probably referring to the fact that a certain site is expected to enroll a few patients. I guess that's what the question is about. So there is a certain expectation that one or more sites will enroll very few patients, whereas maybe other sites will not have that problem and will enroll many patients. But I would say yes. One can always combine sites if this expectation gets to a point where a certain site is expected to enroll very few patients, let's say one. there can be a combination of sites, and that's what is often done in studies that use geographic region as a stratification factor, as the example that I gave. So if worst comes to worst, one can do that. But if the expectation is not so extreme as the one I just mentioned, yes, we would still favor minimization.
Yeah, and I would add a little bit more to that. I would say this is clearly an example where we would not be able to use stratification because we would not have any way to stratify by site. But if we have the minimization can take into account the site as well as other factors. And at the level of the minimization itself, we can use site in the program, and then we can combine several sites into a region. So that's actually, for me, that's a very good example of where minimization is a good way to move ahead. And we just need to then also ensure that the overall balance in the study is also taken into account.
All right, so our next question here is, how can we consider randomized trials in orphan diseases?
Well, it's really a good question. Of course, the logic is the same regardless of whether the disease is rare or not. So ideally, we should have randomized trials. But of course, practically, it may not be feasible. So if this is really the case, one may not be able to consider randomizing. But if possible, we would say always randomize at some point in the drug development process, unless of course it's an extreme situation where there is absolutely no standard of care, the disease is really rare, it's even considered unethical to randomize, the new drug is so promising that very early results are completely outstanding. So clearly there are situations where randomization can be skipped, so to speak, and we discussed that in the paper that I referred to. But in most situations, even for rare diseases, we would recommend as often as possible using randomization.
All right, so our next question here is it possible to justify equipoise in a randomized trial of a highly targeted therapy for a disease with no further options?
Shall I tackle that one?
Yes.
Yes. Okay, so again, this is very much related, in a way it's related to the previous one, because I guess the point of the question is that if you have a drug that is very targeted, highly targeted to a specific molecular abnormality for a disease where there are no further options in the market, or so it's a discussion. There are many published papers about this. Some people are on one side of the debate, others defend the opposite. Again, I would say as much as possible, we like to randomize. Equipoise, of course, is underlying all the randomization exercise, and there are ways to justify equipoise, at least from the point of view of what's called clinical equipoise, which is even though an individual may not be convinced, there is enough evidence for the scientific community that we really don't know, the drug is new, it might be toxic. So even though it's promising, some people would argue we have to randomize from the very first patient in a clinical development program. So I guess it's possible to justify equipoise, although again, I would say in very selected situations of very promising drugs without approved therapies, very rare diseases, one might consider foregoing randomized studies.
All right, we have enough time for one more question here. Can the sponsor have access to the monthly checks performed by the statistician in an unblinded trial?
Well, it comes back to what I mentioned before about the kit list and an unblinded trial. Of course, if the trial is unblinded, the sponsor knows on an individual patient basis who received what treatment. But ideally, we still prefer to keep any summaries of any information blinded to the sponsor while the study is going on. So we would recommend against that. Now, it could be that at the sponsor, of course, if there is somebody in charge of the drug supply and they want to make sure that things are happening correctly, even for a blinded study, an unblinded person at the sponsor would be able to have access to that information. But in general, the key people at the sponsor shouldn't have more access than they need.
All right, well, thank you so much for those answers. We've reached the end of the Q&A portion of this webinar. And if your question wasn't attended during the Q&A session, the team from IDDI will do their best to follow up with you after the webinar. If you have any further questions, please direct them to the email address displayed on your screen right now. Thank you everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Dr. Everardo Saad and Ms. Linda Danielson, by clicking the link I've sent to all participants through the chat box. We hope you found this conference informative. Have a great day, everyone.